To be able to do DNA analyses of old material, whether it's museum specimens, whether it's subfossil bones, whether it's archaeological artifacts, that gives us a window to the past that we wouldn't have had, a way of looking at what was going on to these populations in the past. And a lot of people have found it's extremely useful to take and infer from the DNA what the population processes were, both at the time this was happening, the changes were happening caused by humans, but also even before that point. So DNA gives you a good record of what's happened to populations over different time frames. I'm Rob Fleischer, and I'm a senior scientist and the center head for the Center for Conservation and Evolutionary Genetics, which is part of the Smithsonian Conservation Biology Institute at the National Zoo.
And I'm Jesus Maldonado. I'm a research geneticist at the Center for Conservation and Evolutionary Genetics. Recently, with the advent of this genomic revolution and the development of new technologies for sequencing, it's called high throughput sequencing. We can obtain massive amounts of information from museum specimens and from archaeological remains. And so very recently, we started working with archaeologists here at the museum, the head of the chair of the Department of Anthropology, Dr. Torben Rick, who specializes on Channel Island and Chumash Indian work in anthropology. We started to apply these new novel technologies to get genomic information from samples that were collected in the archaeological record. And so archaeogenomics really is a way to put together various fields, including an archaeologist, conservation genomicist, wildlife managers, ecologists, and try to understand how organisms and humans have impacted organisms in their environment through time, and use that information for conservation purposes. In the past, what we used to do was use a technique called a polymerase chain reaction, which was just amplifying really short fragments of DNA. We were able to address questions with very small and very little information from the genome. But nowadays, with the advent of these genomic technologies, we're able to get a wide representation of the genome, mainly through through capture methods, capturing specific areas of the genome, targeting a more targeted approach to getting the information. But it's made it so much more effective, efficient, and cost efficient for us to get larger amount of information that will help us more finely address some of the questions we couldn't do in the past, right?
One of the interesting sides to this is that we can view archaeological materials as both something to study in their own right, to try to help to understand the human impacts and the human interactions with the environment, but also they represent a sample from many hundreds or thousands of years ago of the animals that lived in the environment at that time, and so we can use those to look at how the populations of these animals have changed over time as well, especially in concert with paleontological material that was essentially there before humans started impacting the species. So it represents a benefit to both fields to do this kind of work, both conservation genomics and also archaeological or archaeology. I'll give you one example from a study we did many years ago in Hawaii, and we're doing additional studies in Hawaii, but this was within the state bird, the Hawaiian goose or nene, and we knew that they'd gone through a bottleneck in the 1900s and the early part of the 20th century, and then recovered. So we looked at genetic variation in modern nene, which was extremely low, and then we started looking back into the past at museum specimens that were collected in the 18 and 1900s, at subfossil bones that were associated with human midden sites and human villages and human settlement. We call those the archaeological samples. And then we also looked at what we call paleontological samples that predated most of the human impacts, were 500 years or older, up to a couple thousand years. And what we found, what was interesting, which we would never have known unless we had done this, is that the variation that was low now, it was also low in the museum specimen samples, and it was also low in the archaeological samples. So apparently there had been a large population decline, probably associated with hunting and other uses from the native Hawaiians after they arrived in the islands, that brought the population of nene down to such a low level that it lost significant amounts of its genetic variability. And it's never recovered that in the time since. So we were able to discover that the bottleneck that occurred was not, well, there was a population decline, another massive one. The original population bottleneck that happened, let's say, 500 to 200 years ago, was the primary reason why the variability was lost.